A man and a woman go to the doctor. They're given the same quality of care and are treated with the same drug, but their outcomes are vastly different. The drug helps improve the man's condition, while it actually proves detrimental to the woman. Why does this happen? The reason is that previous research has ignored biological sex differences, which has led to the development of drugs and therapies that are optimized for males, but sometimes haven't even been tested on female animals. This story happens all too often in diseases called cardiac arrhythmias. Our hearts are electric, and electrical signals allow our hearts to beat rhythmically throughout our lifetime. You can visualize these signals on the surface of the heart by what's called an EKG. You may have seen what a normal EKG looks like before, but when the heart's electrical signal doesn't function properly, this leads to an arrhythmia. And these can have serious consequences like sudden cardiac arrest. Surprisingly, certain arrhythmias are known to occur twice as often in females compared to males. And we hypothesize the reason for this difference is that the male and female heart conducts electricity differently. As you can see from the EKG in the center, females have a longer Q to T interval, shown in red, compared to the male in blue. This basically means that it takes the female heart a longer time to relax after it's contracted. Although this difference has been known since 1910, and we've shown it's conserved in both mice and humans, we don't know why this happens. So to better understand this, our lab identified all of the genes, proteins, and regulatory factors that are expressed differently between male and female hearts. And from this, I found a single small RNA molecule called a microRNA that was expressed much more highly in the female heart compared to the male heart. This microRNA, number 871 specifically, is encoded on the X chromosome, which is interesting because that means there may be more in females because they have two X's, while males have an X and a Y. To understand how this microRNA functions in the heart, we used an inhibitor to reduce its expression. And we found when we did this decreased the female QT interval to now resemble that of the male at baseline. Additionally, when we tested with drugs that are known to cause arrhythmias more often in females than males, this microRNA inhibitor was able to rescue female hearts from developing arrhythmias. This is amazing because this microRNA molecule is only 22 A's, T's, C's, and G's in length, but it can control such a critical sex difference. In the future, we hope to continue this research and apply it to human cells so that hopefully one day it can be applied to patients. In summary, what I hope I've shown you today is that it's critical to better understand biological sex differences so that we can improve women's health outcomes, reduce sex disparities, and provide more equitable care for all. Thank you.